Okay, so let's begin our session today. And hello and welcome to the journal webinar series and Plant Genome Journal. And this is the third webinar of the series, and I'm happy to welcome you all the participants. Please accept my greetings and good morning, good afternoon, good evening, based on the locations you have joined it here for the seminar today. So I'm your moderator and Nilesh Mishra, the social media editor for the Plant Genome Journal. And I thank you all once again for taking time to join us today for this webinar. We are excited to continue this series, which will be held every 2 months and will feature authors and research published in the journals from the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. So before we begin, I'd like to mention that the questions can be entered in the Q&A section, and I'm sure you are able to see that at the middle center of your screen, of the Zoom screen. And yeah, so feel free to write your questions and the queries that you may have around the presentation, and we'll try to address them at the end of the session. Also, please note that the webinar will be recorded and the recording will be emailed to all the attendees. So without any further ado, I'd like to introduce our today's speaker, Dr. Faye, who will be speaking about his research, which shows new evidence that stay green drought tolerance loci, previously characterized from a single drought tolerance donor line, have a broad role in sorghum drought adaptation across Africa. And his research was published in the March 2022 issue of the Plant Genome Journal. So, and Dr. Faye is a crop geneticist at the Regional Center of Excellence on Dry Cereals and Associated Crops in Thiès, Senegal. So with that, I welcome Dr. Faye, and the floor is yours.
Thank you. Hello, good morning, good everyone, good afternoon, everyone, depending on where you are connecting. Thank you for joining this seminar. I'm very glad to be here among you. Today, I'm going to present about our research paper that we published recently, talking about the role of the stay green loci across West and East Africa beyond the original sources of stay green. So I would like to thank, first of all, my colleagues who are involved in this research, and I'm speaking on the behalf of that. So as you know, sorghum is a staple food crop grown over all Africa and can feed about more than 500 million people in Africa. So as you can see in this map on the right-hand side, sorghum is grown across all agroclimatic zones, starting from east to west and through the north-south gradient, rainfall gradient, and photoperiodic gradient. So sorghum is a resilient crop with little agricultural input that are used by smallholder farmers to grow it, low inputs, and drought tolerance or drought stress, which is the major biotic factors that limit its production. So as we know, it's a diploid species and predominantly self-pollinating crops. And the most important things about sorghum is the genome size, which is about 800 megabase pairs. And this makes it a good model species among the grass crops for understanding the genomic of local adaptation, especially drought adaptation. So as you can see in the map here, we have the different type of sorghum grown by smallholder farmers in Africa. We have the corundum types that are mainly grown in the central part of Africa. And Dura types are grown mostly in the eastern part of Africa, while the Guineas are grown in the west and southeast part of Africa. So all these botanical types have some specific traits that characterize their adaptation across the Sub-Saharan African region. So in terms of rainfed smallholder agriculture in the Sahel, sorghum is a predominant crop that can be a food security staple crop. And here we have in the picture, for example, farmers' field in Senegal, where this is well adapted. And if we look at the growing season of sorghum in Senegal, for example, the growing crops occur mostly during the month of July to October. And we have only one growing season for sorghum in Senegal, while we have also two other off-season that the crop might be grown. But in terms of phenotyping, especially drought phenotyping, is kind of challenging, because if we do the phenotyping during the growing season, we might not have the optimal phenotyping for drought. And if we grow it during the off season, either the cool off season, which starts from October to February, the drought phenotyping or the performance of the genotype might not reflect the production environment or the growing season where sorghum is mostly grown by smallholder farmers. So this photoperiodic flowering adaptation is useful for drought adaptation, because the crops is able to mature right at the end of the growing season, depending on the agroclimatic zones across the Sahelian zone. So in terms of drought tolerance, most of the breeding programs in the regions have one common goal, which is to develop drought-tolerant varieties for the smallholder farmers. And this, we know, for example, drought phenotyping or drought selection is challenging, especially for small and young breeding programs, because it requires a year-to-year variation or multi-site evaluation. And this is not quite possible for these breeding programs. For example, in the year 2018, we observed about 1.5 months of drought, and this is quite challenging for the breeders and the smallholder farmers in general. So the research goal to tackle this is to identify some drought resilience genes that can be used for marker-assisted selection. And the hypothesis that goes with this research goal is that the oligogenic architecture for drought adaptation, underlying drought adaptation, and this has some literature backups, because we know, for example, the stay green drought tolerance, which is the capacity of the plant to maintain its leaves after the possible the flowering time, has been demonstrated by many programs. And given also the nature of sorghum, especially the land races grown by smallholder farmers in the region, we might expect that some major effect loci can be selected either by farmers or natural selection, and can be identified through the new technology of gene mapping. So the overall goal for this research was to discover and deploy drought tolerance trait by the year 2025. And the ultimate goal are two. First is to determine the genetic architecture of drought tolerance, and then develop varieties that will be drought tolerant and can be used by smallholder farmers. But we started with farmers' knowledge that are highlighted here with the green color, the breeders' knowledge and resources highlighted with pink color here, and the knowledge generated by the global scientific communities highlighted in pink color. So based on farmers' knowledge and farmers' preferred varieties, some participatory rural appraisal have been carried out by breeders, geneticists, and physiologists to gather all the farmers' preferences, the local varieties for different agroclimatic zones across the West African region. So this germplasm, in addition to it, breeders, physiologists, and geneticists assembled also breeding lines for different programs and assembled to make a panel that were phenotyped of drought stress environments. The same panel has also been genotyped, and the genotype and phenotypic data were used to identify loci that would be or would underline drought tolerance. So taking advantage also of the global scientific community's generated knowledge regarding drought tolerance. The QTL that are identified from this research have been or currently being validated using the cross that were developed by the breeders based on the varieties that show performance under drought stress conditions. So CASP markers have been developed and currently being used for multi-acid selection to follow the lines that will carry the favorable alleles for drought adaptations. So the lines carrying these drought adaptation loci will be evaluated on research station and later on the farmer's field in order to, first of all, understand the genetic architecture of drought tolerance, and then identify also drought tolerance or develop drought tolerance varieties for the different stakeholders. So what is known about drought tolerance or drought adaptation? We know, for example, stay green trait has been characterized, well characterized, especially in the Australian germplasm. And a key stay green tolerant varieties is the BTX642, formerly known as B35. This is an Ethiopian-derived Durak varieties that is known for tolerance to post-flowering drought stress conditions. Beside these lines, we have a TX7000, which is a classical US varieties, but in the contrast to BTX642, TX7000 is a drought susceptible varieties. So these are international drought reference check lines that help us to identify or precisely phenotype drought tolerance over the environments in West Africa. So we know also stay green traits involve several aspects of the plant development, involving, for example, water use efficiency, capacity of the plant to extract water from the soil, leading to non-senescence plants, and later yield increase or yield stability. All these are knowledge that are available through the literature, and we took advantage of this knowledge to undertake a drought experiment over the region. So we know also based on research about drought tolerance that stay green loci exists in the materials, especially in the US and Australian germplasm. And several QTLs have been identified from different genetic backgrounds. Here shown in this figure, for example, we have the 10 chromosome of sorghum on the x-axis, and on the y-axis, we have the different QTLs that have been mapped in different germplasms. So this is an important resource as it is available and can be used, and we took advantage of these resources to identify drought tolerance genes. But the challenging aspect here is that we don't have for sorghum any genes that have been cloned for these stay green QTLs yet. And so the idea is to identify and further in the pipeline, try to identify genes that will have functional variation that can be used for drought improvement in the region. So coming back to the hypothesis, we hypothesized that oligogenic architecture underlying drought adaptation in West Africa. And this hypothesis we subdivide into sub-hypotheses. The first hypothesis is that stay green alleles exist in West African germplasm. We know, for example, stay green already exists in the Ethiopian germplasm, given the nature of B35, which is a post-flowering drought tolerance. But we know also that Duracodatums that are hypothesized to be drought tolerance diffuse from Eastern Africa, especially from Ethiopia, across the Saharan region toward West Africa. So we hypothesize that those same stay green alleles would exist also in the West African sorghum germplasm. Another competing hypothesis for this is that in addition to the known stay green alleles, novel drought tolerance or avoidance alleles might exist also in the germplasm that are different from the known or the canonical stay green loci. So coming to the research, we assembled the germplasm local varieties predominantly and breeding lines from 4 national programs in West Africa, listed here in the map, from Senegal, for example, Mali, Niger, and Togo. And if we look at This germplasm covers most of the agroclimatic variation from north to south gradient, including the photoperiodic gradient. So this panel, we name it as WASAP, or the name West African Sorghum Association panel. And it consists of 750 accessions that predominantly are land races and most of the important breeding line for the different national programs. So this collection has been characterized using genotyping by sequencing of 160,000 SNP markers, and genetic characterization has been carried out. The most important part of this analysis is that we identified 8 ancestral subpopulation. And the good part of this finding is that most of the ancestral population identified germplasm exists in all the 4 programs. There are a few ancestral populations that are unique to a given program. For example, the one we have here in this color are only specific to the Niger germplasm, while these ancestral population in forest green are unique to the Togo germplasm, which is in the highly rainfall areas across the West African region. So we demonstrated that in 2021, that these resources are useful for identifying alleles that underlie agronomic trait of importance. So we demonstrated that this resource will be suitable, for example, for genome-wide association study and to dissect the genetic architecture of drought tolerance in this collection. So the challenge of carrying drought phenotyping, we know for all, there is a confounding effect between flowering time or photoperiodic flowering time and drought adaptation itself. So for example, we have the main raining season that occurs from July to October. And it is hard to impose or to imply what is stress during the growing season because we cannot control the rainfall. And if we do the phenotyping during the cool off season, which is start from October to February, the performance of the genotype might be different as well from the performance under the growing season. So this brings the question whether the off-season drought phenotyping will reflect the growing season or the production environment of sorghum. The third challenge aspect is that if we do the phenotyping in the hot season, which occurs between March to July, because of the photoperiodic flowering time that characterize most of the germplasm, some of them will not even flower before reaching to the critical photoperiod time, which is September 21st. Or beyond that, the data we observe or we collect might not reflect the growing season or the production environment of sorghum in this region. So these are the challenges for the phenotyping that we carried out. And the hypothesis that local adaptation underlying drought adaptation is confounded by the photoperiodic flowering time differences that might challenge the genes or the QTL that will be identified based on the phenotyping of drought. So we undertake or carried out large-scale managed drought stress environment phenotyping based at the research station at Bambey. Bambey is part of the Saharan zone in Senegal. And if you look at, for example, the red dot here, this research station or the position of this station reflect most of the Saharan zone from west to eastern Africa. So the two pictures on the bottom reflect or shows the different experiments on the well-watered, which is the control environment, and the water stress environment at the bottom. So field phenotyping was done using the 750 accessions, and we use augmented block design with highly replicated checks, including the 2 international drought reference check lines. So in addition to that, also, we make sure that each plot was surrounded by an alternate check row to control the edge effect. And for the post-flowering drought stress evaluation, the germplasm was divided into 3 maturity groups in order to effectively imply or impose the water stress in the post-flowering drought environment. So we have a phenotype over 4 years, starting in 2014 to 2017, where in 2014 we had rain-fed condition environment, where the sowing date started in August in order to have a post-flowering drought or simulate the post-flowering drought as it occurs during the real growing season of sorghum. In the off-season, in 2015, 2016, and 2017, we had a well-watered control environment, stated here as WW. WS1 is the pre-flowering post-drought stress, and WS2 is the post-flowering drought stress. So this was done with the collaboration of our colleagues in Senegal, Bassirou Sine, ecophysiologist, and Jean Agata, a breeder from Togo, who were based at Seras in Senegal. So just to show an example of the different treatment that we have, for example, on the top left corner, we have the control environment. The bottom and left-hand side, we have the pre and the post-flowering drought stress environment. So we have on the right-hand side, for example, one of the check B35, which is a post-flowering drought tolerance variety. And we can clearly see, for example, the difference in terms of greenness between this check varieties and the other germplasm that were used in this evaluation. So to demonstrate that the water stress, drought stress that we imposed was effectively assessed, we took measurement about the fraction of transpirable soil moisture or soil water expressed in this y-axis, while on the x-axis we have the day after the planting. So we have, for example, on the top here, the well-watered where the plants were fully irrigated till physiological maturity. In the WS1 or the pre-flowering drought stress, the plants were fully irrigated until 30 days after planting, and water stress was imposed for 1 month, and then the plants were rewatered again. In the post-flowering drought stress, WS2, 3 maturity group, depending on the maturity group, the water stress was imposed as well. So what we can see here, for example, in blue, we have the well-watered, or the fraction of transpirable soil water in the well-watered experiment. We see a stagnant curve where the stress or the irrigation was fully applied, compared to, for example, in the pre-flowering environments, where we see, for example, the dropping of the water availability in the experiment. And in red, we have the post-flowering soil moisture, which you clearly see that we were able to precisely control the water stress in the different experiments. In addition to demonstrating that the water stress was successfully imposed, we measured also the performance of the different genotypes or varieties by using the 1:1 ratio. For example, we have on the x-axis the grain weight on the post-flowering drought stress environment, and on the y-axis we have the grain weight per plant on the pre-flowering drought stress environment. So if we consider, for example, the 2 reference check, which is TX7000, the successful drought tolerance, post-flowering drought tolerance, and in red, the B35, the tolerant post-flowering drought tolerance line, we see a clear cross genotype environment here, for example. So we see B35, for example, will perform better than TX7000 under post-flowering drought environment, where the TX7000 outperform B35 under the pre-flowering drought. And so this demonstrates that in addition to controlling the water supply, performance of the genotype demonstrate that drought were considerably sensed by the different plants or genotypes. So if we, for example, we consider the different botanical types, because we know in sorghum, for example, Dura types are more tolerant to drought compared to Guinea, for example, that are more adapted to the higher rainfall areas. And so we expect to observe a higher performance in terms of grain weight in the Dura types compared to the Guinea. However, what we observe here, we didn't see any difference between the different botanical types, especially Guinea and Duras. But what we can see, for example, the intermediate types between the Dura and the Codatums, which are referred here as Dura Codatum, they perform better than their basic botanical types. And what we can observe here, for example, also, is that the post-flowering drought environments reflect better the growing season or the rain-fed condition environment. As we see, there is no significant difference between these two water regimes. However, if we take or we consider the pre-flowering drought stress environment, there was a huge reduction in grain weight per plant compared to the production environment or the rain-fed drought stress conditions. So coming to the identification of loci that may underlie drought tolerance in the germplasm, we carried out genome-wide association studies. So the figure we have here represent the Manhattan plot, where we have the 10 chromosomes on the x-axis, and on the y-axis we have the significance level of the association. So the red dots indicate markers or SNPs that are significantly associated with the trait. And here the trait that we are showing is the stress tolerance index that characterizes or shows the capacity or the level of tolerance while maintaining higher production for this germplasm. So we highlight also the canonical stay green QTL that have been mapped by several studies. And in the RUG bar plot in red here, we are showing the positively pleiotropic marker trait association. So here, positive pleiotropic, we mean, for example, if the QTL or the association have been identified in both pre and post flowering drought stress. And we see, for example, these positive pleiotropic drought QTL, they colocalize with the known stay green loci. In addition to that, the most significant association were not colocalizing with the known stay green QTLs. And so this has a huge impact because regarding or coming back to our two sub-hypotheses, whether the West African germplasm harbor stay green alleles or not, or novel alleles for drought tolerance can clearly be observed through these GWAS studies, for example. We identified novel loci that have not been identified yet. In addition to that, we were able to show that the West African germplasm harbor alleles that colocalize with stay green, known stay green QTLs. And so we are in the process of developing markers and validating these markers and using these markers for the integration of drought tolerance in the elite germplasm. So another question that we asked was that, do these drought tolerance QTL or loci underlie drought adaptation? So here is a figure showing the pi ratio in terms of pairwise nucleotide diversity reduction relative to the wild species or the wild relative of sorghum. So on the top panel, we have the pi ratio in the Dura Codatum genome, which we showed previously that this Dura Codatum, they perform better under both conditions, pre and post flowering, compared to the production environment. And on the figure, the panel on the bottom, we have the Dura or the pi ratio in the Dura genome. So we were able to show as well, based on the enrichment analysis that we performed, a perfect colocalization of both the positive pleiotropic QTLs and the stay green QTLs. And also in region where there is a reduction of pairwise nucleotide diversity, which is a synonym of positive selection in the genome, either in dura cordatum or dura genome. And we went beyond this observation because in part of testing the hypothesis whether the drought tolerance or the stay green allele exists in the West African germplasm, we have these two competing hypotheses. For example, one, West African drought tolerance QTL in stay green alleles originate from spread of the stay green QTLs. And the second hypothesis is that these alleles do not originate from the spread of the canonical stay green alleles. In other words, they are novel or different. And one of the results that we are showing here, for example, especially one of the associations that colocalize with stay green 3 here, we have observed that the alleles at this association exist in both Eastern Africa, where the stay green was firstly identified or found, as well in the West African sorghum germplasm. Eastern germplasm. As we see here in the map, each point represents the geographic origin of a sorghum genotype or sorghum line. And so we didn't clearly find or observe a result showing that these alleles are the same. However, some functional genomics are needed, especially cloning the different QTLs here, in order to see whether these alleles are the same or they are different. But what we observe is that some novel alleles or QTLs have been identified in the West African germplasm that have not been identified somewhere yet, in addition to the known stay green QTLs. So in the road of discovering and deploying drought tolerance trait, this is where we are right now. So crosses have been made based on the putative drought tolerance lines crossed together, and the validation of the QTL is currently being generated. So we develop markers and we follow the integration of the drought tolerance QTLs in those crosses in order to undertake a marker selection and test the lines that carry the favorable alleles in both research station and farmers field. So the goal is first to understand the genetic architecture by combining the knowledge provided by the global scientific communities and the knowledge coming from the farmers field and farmers preferences with what we have tested through this research study. So to just show what we are right now based on the identification of the drought tolerance QTLs, we develop markers and we follow the integration of the favorable alleles into the developed lines. And we have, for example, here in blue is showing the favorable allele at a given QTL. In red is non-favorable allele at a given QTL. And we see, for example, in some of the families that we develop, some of them, they are already fixed for the major favorable alleles. And right now we are evaluating this germplasm in lysometer experiment and research station to evaluate the effect of stay green in the Sahelian region. So this is a promising result that we will be able to deploy drought tolerance into the region, but beyond the region of the Sahel, because this will be useful for the scientific community. And I would like to thank all the participants of this research, especially involved in the SbZfl1 project, colleagues from Sanara, Imran from Niger, Itra, Togo, from Mali, and Ikrizat, Surat, KSU, and Colorado State University. Thank you for listening.
Thank you very much, Jack. Thank you for the very elaborative presentation and sharing the details about the research work that you conducted and was published in the Plant Genome Journal. Let's see if we have any questions from the participants. I once again request the attendees, if they have any questions, please feel free to type into the Q&A box and we'll try to answer them. Okay, so there is a comment from Benjamin Cole, great presentation. I don't have any questions. So that's a compliment for you, Jack. And yeah. So.
Yeah. Thank you, Ben.
Any other question from the participants? So I don't see any other questions. Maybe I have a couple of questions if I can ask. So Jack, during your study, were there any trade-off between stay green, drought tolerance, and the grain yield?
Yes, that's a great question. Trade-off between stay green and drought. Actually, research have shown, for example, if you look at research done by Australia, especially Borel, there is no trade-off that have been shown between the stay green and the grain yield. Because one of the slide I showed, the stay green phenotype occurs right after the flowering time in terms of drought stress, when drought occurs. And once the stay green phenotype is involved, the plant develops some mechanisms to better extract the water available in the soil, but also to maintain water available in the plant itself. And so during all the grain filling time, the plant has the resources to accomplish the grain filling until the physiological maturity stage. So based on the research, there is no trade-off between the stay green and the grain yield. It either maintains the yield stability or increase the yield depending on the level of the stress. And we did observe this also as well, because lines that harbor the favorable alleles, most of the time they outperform the other genotype that doesn't have the favorable alleles in this germplasm.
Thank you. Thanks, Jack. And I see there are a couple of other questions from the audience. Cosmos say that excellent presentation. Any plans to extend this work to Southern Africa?
Any plan to extend this work? I'm glad, Cosmos, you are finding some interest in this work. For sure, this stay green has been found previously in Ethiopian germplasm. And we know, for example, there has been a diffusion from Ethiopia to other parts across Africa, either west or southeastern part across Africa. And probably there might be some genotypes or varieties, especially local varieties, that will harbor the same stay green QTLs as we just did in the West African germplasm. And so it will be more useful if the local germplasm have the stay green QTLs, because those they are not exotic, especially in breeding, for example. Instead of using B35, which is the well-known stay green drought tolerance line, if you have in your local germplasm a line that has the same QTLs, it will be easier to use that line and make process to improve the local germplasm over there, instead of using an exotic material where only maybe you are interested in the stay green, not other agronomic traits. Because we know, for example, the example or the case we observed in the matter here in West Africa, except the stay green trait from B35, most of the other agronomic traits are not important because of the stature of the plant in terms of height, the nature of the panicle, and so many other agronomic traits. So yes, I fully support that extending this work across other regions will improve the scientific knowledge and develop better varieties for drought tolerance.
Great, thanks, Jack. I hope that answers Cosmo's question. And yeah, so another question is from Balua Nebe, and I guess correct, his colleague from ICRISAT, and he says, congratulations, Jeff and team, great results. Do you have any preliminary results on the stability of these QTLs across different environment?
That's a great question. Thank you, Balua. We are just starting testing the introgress line, the line that we developed by introgressing the stay green QTL in to the elite varieties. We haven't got the result regarding like mini environment, for example, in Senegal, for example. Maybe in the coming year, we will have some data. But right now, the experiment that we are just carrying out is only done in Bombay and is representing only one environment. So we don't have a result for that yet. But it's a great question. It would be interesting to evaluate this line across multiple environments. And for example, I had a discussion with Santosh from ICRISAT. I think he's one of your colleagues. And they use stay green also to improve for drought tolerance. And based on the evaluation, among the 4 known stay green QTLs, only stay green 3B especially, were stable across most of the environments that they were tested. But maybe the environments might be different, so we will see.
Yes, so great. So there is a response from Balua. Thanks, Jack. We'll be happy to support this work.
Yes. That will be a great opportunity to test that. Thank you.
Yeah, good. So I don't see any more questions on this. And just for the interest of the participants, if you want to read more about the paper, the link has been shared in the chat. So you can access the paper online, open access. So yeah, and well, with that, I don't see any more questions. So thank all of you for joining for today's webinar, and thanks to you, Jack, very much for taking time to share your work with the participants.
Yes, my pleasure.
Yeah, and yeah, just for a reminder to the participant, the recording of this session will be made available to all the attendees and will be available on the YouTube channel as well. So with that, thank you all. Thanks for joining with us today, and have a good day. Bye-bye.